Question: what is simple staining? Answer: simple staining is a technique that uses a single stain or dye to color all cells or microorganisms, allowing for easy observation of cell shape, size, and arrangement. Question: what is dye? Name a few dyes used in microbiology. Answer: dyes are the crude coloring agents. These are of two types, natural and synthetic ones. For example, crystal violet and safranin. Question: what is the difference between stain and dye? Answer: dyes are the crude coloring agents used for general purposes, whereas stains are the purified preparations. Question: define the dye. Answer: dyes contain both chromophore and oxochrome groups to benzene rings. The chromophore group provides the compounds the property of color, hence called chromogens. It is not dye. The dye should contain an auxiliary group, i.e., oxophore. Its fixation of oxochrome group is to furnish salt-forming properties to the compound. Question: what are acidic and basic chromophores? Answer: the acidic chromophore includes the nitro group, quininoid group, whereas basic has azo, the azine group, and indamine group. Question: name a few acidic dyes. Answer: acidic dyes, such as eosin and acid fuchsin, are anionic, meaning they carry a negative charge. When these dyes ionize in solution, the dye molecules acquire a negative charge. This negative charge allows them to bind to positively charged components of cells, such as proteins in the cytoplasm or extracellular matrix, making these structures visible under a microscope. Question: give some examples of basic dyes. Answer: basic dyes, such as safranin and methylene blue, are cationic, meaning they carry a positive charge. When these dyes ionize in solution, the dye molecules acquire a positive charge. This positive charge allows them to bind to negatively charged components of cells, such as nucleic acids and cell membranes, enhancing the contrast and making these structures more visible under a microscope. Question: what do you understand by mordant? Give an example. Answer: a mordant is a substance used in staining techniques to fix or intensify the color of a dye by forming a complex with the dye and the target tissue or cell. It helps to improve the binding of the dye to the specimen, ensuring that the color remains stable and does not wash out during subsequent processing. Example: iodine. In gram staining, iodine acts as a mordant. It forms a complex with the crystal violet dye inside the bacterial cells, creating a crystal violet iodine complex that is retained by gram-positive bacteria during the decolorization step. Question: explain the method of preparations of bacterial smear. Answer: clean the slide. Ensure the slide is clean and dry. Add a drop of water. Place a small drop of sterile water on the slide if using solid culture. Add bacteria. Transfer a small amount of bacterial culture onto the drop of water. Spread the sample. Use a sterile loop or another slide to spread the bacteria evenly. Air dry. Allow the smear to dry completely at room temperature. Heat fix. Pass the dried slide briefly through the flame to fix the bacteria to the slide. Proceed with staining. Apply the stain as needed for microscopic examination. Question: what is its mechanism of staining? Answer: the mechanism of staining involves dye penetration. The dye enters the cell. Binding. The dye binds to specific cellular components based on chemical properties. Coloration. The bound dye imparts color to the targeted structures, enhancing visibility. Differentiation. Multiple dyes can be used to differentiate between cell types or structures. Fixation. Heat or chemical fixation preserves cell structure and kills microorganisms. Question: what is the purpose of simple staining? Answer: the purpose of simple staining is to enhance the contrast between microorganisms and their background, making them more visible under a microscope. Question: what information can be obtained from simple staining? Answer: simple staining provides information about the morphology, shape, size, and arrangement of cells. Question: how does a basic dye work in simple staining? Answer: a basic dye, which has a positive charge, binds to the negatively charged components of the cell, such as nucleic acids and cell membranes, staining the cell. Question: what is the most common mistake to avoid during simple staining? Answer: a common mistake is applying too much dye or not rinsing properly, which can lead to overstaining and make details difficult to see. Question: how long should the dye typically be left on the sample in a simple staining procedure? Answer: the dye is usually left on the sample for about 30 seconds to 1 minute. Question: why is heat fixing important before applying the stain in simple staining? Answer: heat fixing adheres the microorganisms to the slide, kills the bacteria, and preserves their shape for better observation. Question: can simple staining differentiate between different types of bacteria? Answer: no. Simple staining cannot differentiate between different types of bacteria. It only highlights basic cell structures. Question: what is differential staining? Answer: differential staining is a method that uses multiple stains to differentiate between various types of cells or structures based on their staining properties. Example: gram staining differentiates bacteria into gram-positive, purple, and gram-negative, pink, based on cell wall characteristics.